All right, everyone can unmute. Welcome to the Evidence Reasons and Academy. This is the place to get bored and tortured with nerd stuff. And Emory and I are just gonna have at it tonight. We're gonna get like the nerd nexus here. So what's Rebecca doing here? Well, she's a nerd, at least an honorary nerd, if not a full-blown nerd. She has another stream, and so she's graced us with her presence. And I just invited her to just kind of just say hi. And Rebecca, if you have to move on, it's just I just wanted to have you on my stream for just a little bit. So
cool. Thanks for inviting me, Sal. And I hope you guys have a great torture session. Enjoy.
All righty, take care.
Thanks.
All right, Emory. We have
Now the fun starts.
now the fun starts. Okay. And
now we get to torture each other and not have to drag Rebecca through that.
yeah. All right. So why don't you introduce yourself while I try to cue some things up here for us?
Sure. My name is Emory Moyna. That's obviously a pseudonym in case you haven't figured that out by now. I have a website, inhisimage.blog, where I write about creation stuff, and I've got now 2 YouTube channels because one wasn't enough. And so I've got one where I just talk about biology, more of a nerd style type stuff, where we're talking about issues related to creation biology, and that's the logo that you see on the screen currently, creation biology. And for those of you who remember me as In His Image, that's still there. The channel's still there, and I'm still posting to that, but there's not going to be any science content there. That's all going to be social issues. So I've split the 2 out just to make it easy in case somebody doesn't really want to deal with any of the social issues or doesn't want to deal with the nerd stuff. They still have content they can look at and enjoy.
And I'm Salvador Cordova. I'm a molecular biophysics researcher, and I have 5 degrees, but not a PhD. I have MS in applied physics and a MS equivalent in biology, MS from applied physics from Johns Hopkins University, which means a lot to me. That was the hardest thing I ever did in my whole life. Oh my goodness. And Emory is now one of us now. He is not just a graduate student, he's a graduate student. Master of Science in Biology. Congratulations, sir. God bless you. Proud of you. Top of his class, I presume, or very close to it. And it shows that creationists can do well at biology despite what they say. And I actually think down the road, if they just let things work the way they're supposed to. We lost Emory. He's back. If we let things work the way they're supposed to, I actually think the creationist viewpoint will actually make better biology, because we will look at things and we might actually explore a topic just like that. We will highlight how evolutionary biologists have messed up the understanding of operational biology. We'll talk about that a little bit tonight. And then, since Emory is a special guest, whatever is on your heart, brother, that you want to pour out on for nerd stuff, I'm here at your service. It's just great hanging out with you. And I can't believe it. We got an audience of 7. This is scary.
I don't know. 7 people want to see us torture ourselves, Sal. I'm not so sure about this.
I have, honestly, I was proud. I've had shows where there was no one in the audience, and I was happy. And I'll tell you why. It's not because, I love people, but, I feel like I need to read every comment and, at least say thank you or something. And it takes at least half an hour just for my few little videos. And if my channel grows, I'm going to be ignoring all these people because I just can't manage it,
Yeah.
We
aren't full-time streamers. We can't spend time, we can't spend time managing an audience of any length of size because we have, jobs and lives, or at least we pretend we have lives. We're nerds. We don't have lives. But, we have other things we do, so we couldn't manage big audiences, which it makes me grateful for the fact that I don't really have a huge audience. I think I've between the two channels, I don't think I have 100. I don't think I have 10 people, so it's not too bad at the moment.
And let me see. The banners that I use, it causes some problems on StreamYard, but I'm gonna stream it. I'm gonna just let this banner. Let me see if I could get the ticker to roll. Oh my, look at that. I'm not gonna comment very much publicly on that, except this. I need to put that in the video description. Hang on.
I think it's in the video description, Sal.
I didn't see it there. They're actually now. They're going to be. I expect there's going to be more articles on this. But suffice to say, there's God willing, there's going to be more coming. And that's all I feel comfortable talking about right now. But since people drop in and they're curious what's going on, because I've mentioned this so much on my main channel, there is another channel, Evidence and Reasons for the Christian Faith, which is not quite as nerd intensive. Hang on, I'm amending the video description. Okay. So if you want to access it, it's there. It's there. So tonight, just to open up how we got into all this, is I was teaching Cindy Lincoln some molecular biology, and our mutual friend, Dr. Joel, was in the side chat, and he's saying, I said, we're trying to state the central dogma. And since you're a graduate student, I'm just going to put you on the spot. Can you give an approximation for what the central dogma of molecular biology is?
Central dogma is DNA to RNA to protein.
There you go. Dang. And there's so many statements of the central dogma of molecular biology, and everyone will say, no, that's not right. That's not right. That's not right. And there's not, it's not really important to get even though dogmas and creeds are supposed to be, every word's supposed to be just right. This is biology. So I was trying to teach Cindy Lincoln, and I said, how about this? It's approximately some DNAs transcribed to RNAs and some of those RNAs transcribed to proteins. I said, you'll get yourself out of trouble if you use those.
Yeah, because if you, so I quoted the dogma, but that doesn't mean I believe the dogma. So it turns out you can go back. I know you can go backwards from RNA to DNA, and I'm fairly convinced that you can go backwards from proteins to RNA, and I'm fairly certain you can also cut out the middleman. So,
whoa! All right, I cut you off. Go ahead, Emory. I'm sorry.
So the dogma is a nice general explanation, but it doesn't hold for all cases.
Whoa. Okay, you dropped some interesting. All right, we'll just biology can do all sorts of amazing things, so I'll just leave it at that. Yeah. But anyway, Dr. Joel weighed in. He said, actually, practically all DNA is transcribed. And it's like, whoa. He said, didn't you? He said that's an encode. And I'm like, oh, I thought you'd be the last guy that'd be citing encode. And I said, okay. And I said, if that's the case, I said, well, how about telomeric and centromeric RNA? And he said, he said, well, it transcribes doesn't mean it's useful for anything. And I said, you know what? Why don't I just Google around?
Yeah. Generally, when an evolutionist makes a claim that I'm not so sure about that, the first thing I do is go to Google Scholar and Google it.
I'm exaggerating here, obviously, just so take it for what it's worth. If you hear what an evolutionist says, if you accept the opposite, you have a good chance of being right.
Yeah.
Okay, I'm exaggerating, but you get my drift, I think. So, were you aware of telomeric RNAs?
I think at some point they were vaguely mentioned in a genetics class that I took, but it's been a long time since I've even thought about those.
Okay, so I'm actually going to need your help.
Okay.
You know, I'm a protein guy. I'm not
Okay.
a genomics guy, and that's
Okay.
that was kind of an odd relationship because my boss John Sanford
Yeah, he's a gene guy.
Was a gene guy, and I'm like proteins. So let me just monitor the comment section here, and all right, I don't see anything there. So let me show my screen. Let's do a screen share here. So if you see me looking over here, it's because I have a screen that I wanted to share.
Mm-hmm.
And I'll make this bigger. So
I'm gonna go
from the prestigious scientific journal Nature, telomeric.
That's a puny podunk journal, so nobody cares about that.
Yeah. Telomeric RNAs are essential to maintain telomeres. Telomeres are transcribed, generating long non-coding RNAs known as tera. Deciphering. Oh, we got it. I got to see this now. Deciphering the role of tera has been one of the unsolved issues of telomere biology in the past decade. So, okay, Dr. Joel said they're all transcribed, but it doesn't mean that it has function, and that kind of got me going. He said, yeah, I think those are transcribed. So this is just kind of a little thing to, here's one for the creationists here. If you have any thoughts so far, want me to just read the abstract?
Right off the hop, I'm looking at this and going, oh boy. So they're essential to maintain telomeres. Well, that means they actually do something.
They actually do something. Take that, Larry Moran. Take that, Dan Grauer, you evolutionary biologists and apologists. Ha!
I have to admit, I used Dan Grauer's textbook for one of my genetics classes, and there were several times where the professor would make little offhand comments. Like, she wasn't a creationist, she was an evolutionist, but she'd make little offhand comments about how he was out of step with the majority view in certain places because he'd make snide comments about encode and gene functionality and all this other stuff. It's like, and she'd just kind of go, yeah, Grauer thinks that. That's not what the literature says, but Grauer thinks that. It's like, okay.
Yeah, evolutionary biologists they're kind of out of step, I mean, that's just, a lot of people, it's like, oh, evolutionary biology is the central thing of biology. And it's like, well, if it is, how come they keep mischaracterizing it and can't get the basics right,
Mm-hmm.
So sorry, maybe we can just dispense with it. Anyway.
Yep.
You know what? You are the man to read stuff. Would you be at all interested in reading through this, or did you have something else on your heart?
I can read through it. I don't have anything. I mean, there are a few things I could always talk about. There's always things I'm interested in talking about, but this looks interesting. So I probably won't know all the terms, but I can at least have a look at it, and we'll see where we go.
Well, hey, maybe we can have some fun just poking at the evolutionary biologists out there, especially. Oh, and there is Dr. Joel speak of the devil. Ha ha.
That wasn't very nice, Sal.
Oh, I'm sorry.
I know what you meant, and I think he does too. All right, this is interesting.
All right, okay. So, thank you for gracing us with your presence, Dr. Joel. Let's see here. Okay, guys. I don't know why we get these obnoxious advertisements. This is how I guess even Nature will be a little bit of a cheapskate and make advertisement money. See if I can turn this.
Sal, you need to run ad block.
Oh, that's right. I happen to have an ad blocker. One moment. Let me activate it. Hang on. Why didn't I see? That's why I need you, Emory. You kind of got my back here, and you get everything working nice. So, okay, accept all cookies. See, I forget ad block also works for those obnoxious text things. So I can scroll through this while you don't have to read it from my screen unless you want. I mean, you know what I'm showing. All right. So do you want to go ahead and read, Emory?
Yeah, I can read. Although you may want to read Dr. Duff's comments before we get into that, in case you want to. Go
Okay. Okay, to be fair, why is this surprising? Got no problem with telomeric DNA being transcribed and used to regulate telomeres. My point was, most of the genome is transcribed. I made a video about random DNA being transcribed. Thank you. All right, so here we go. You can start from the start. How about the title? Title is
Telomeric RNAs are essential to maintain telomeres. And this is from 2016. So this is a 6.5-year-old article at this point, something like that.
Getting to be 7. Yeah, 6.5. Yep.
Sheesh. Abstract. Telomeres are transcribed, generating long non-coding RNAs known as TERA. Okay, I'm not sure why they decided to put an extra abbreviation in there, but okay. Deciphering the role of TERA has been one of the TERA, hang on. Is that telomeric? Oh, I don't know. I'm sure it means something. Deciphering the role of TERA has been one of the unsolved issues of telomere biology in the past decade. This has been in part due to a lack of knowledge on the TERA loci, thus preventing functional genetic studies. Here we describe that long non-coding RNAs with TERA features are transcribed from the human 20q and XP subtelomeres. Those I'm not familiar with. Sal, do you know what those are? I'm not familiar with those are.
No, this is a good. Okay, this is why we like going through peer-reviewed things. So maybe we can learn what these terms here are.
Yeah.
I'm embarrassed to say this, guys. Like I said, I'm not a gene guy. I am a protein guy. But even then, if you're a protein guy, you might specialize in one or two proteins and that's it. I mean, everyone is so specialized. So I don't even know what the word subtelomere means. So you're going to have to, can we finish the abstract and then we can
finish the abstract and we'll come back to the phrases as necessary. All right. So deletion of the 20q locus by using CRISPR-Cas9 caused a dramatic decrease in TERA levels, while deletion of the XP locus does not result in decreased TERA levels. Strikingly, the 20q TERA ablation leads to dramatic loss of telomere sequences and the induction of a massive DNA damage response. These findings identify chromosome 20q as a main TERA locus in human cells and represent the first demonstration in any organism of the essential role of TERA in the maintenance of telomeres.
Wow. How about that?
Yeah, so what it looks like, I'm assuming that the 20q and XP means chromosome 20 and the X chromosome. I'm assuming that's because just reading that because from the last sentence, these findings identify chromosome 20q. So I'm not sure what the Q and P notation is. That I'm not as familiar with.
All right, neither do I. So what is a subtelomere? Shall I bring that up on Wikipedia?
Yeah, bring that up. I think I know what that is. I'm pretty sure I remember what that is, but it's better to pull it up and make sure we actually have the correct definition so that I'm.
Yeah. Oh, here we go. Oh, thank God for Wikipedia. And actually, there really are, depending on who the authors were, it could be great or it could be just awful. Anything on Christianity, intelligent design, or creationism is just awful. Written by Darwinists, not written by people that are actually in the field.
And it's not. And the thing is, it's not written by researchers. It's written by the rabid fundamentalist atheists.
Yep. And you know what? I'm sorry to say this, but given how the creationists don't agree, we would do better, but we'll be fighting amongst each other. So, okay. So subtelomeres are segments of DNA between telomeric caps and chromatin.
All right.
Oh, and here from a biology professor, thank you, Dr. Joel. subtelomeric regions are areas adjacent to telomere repeats.
Okay. Yes. Yes, that makes sense. So
Apparently,
subtelomere is would be below the telomere, which would be the point which would be adjacent to the telomeric repeats. That makes sense. Yeah. Yeah.
so let me see this. Do I have this correct? At the very edge of the chromosome, DNA is the telomere, and then
right at the telomeres are caps on the ends of chromosomes.
Chromosomes.
And so the subtelomeric regions would be regions directly before those caps, which would then regulate the telomeres. It makes sense that they'd be directly there rather than somewhere else on the chromosome.
Okay, so telomeres are protein DNA constructs present at the ends of eukaryotic chromosomes.
Mm-hmm.
Whoa, okay, all right, time out. I didn't know this was a eukaryotic specific feature.
Yes, because most bacterial chromosomes, non-eukaryotic chromosomes are they handle DNA differently in bacteria and other non-eukaryotes than eukaryotes do. So that does not surprise me at all. That I didn't realize the telomeres were unique, but to eukaryotes, but it makes sense that they would be because
Yes.
The difference is that between eukaryotes and prokaryotes is that prokaryotes don't have membrane-bound organelles, and their DNA is held. I'm talking way too fast. Slow down. Their DNA is stored somewhat loosely. Yeah, but you have the circular plastids. So you don't need caps on the ends of those because you're going in a circle.
Dang! This rings of discontinuity systematics. God bless you, Walter Remind.
There are significant differences in the way DNA is handled in prokaryotes versus in eukaryotes, and those differences. I'm sure there are papers. This is not something I have investigated extensively, but I think that is something that would need to be accounted for. And maybe Dr. Duff knows of some papers that deal with this particular topic, how you go from a bacterial DNA arrangement to a eukaryotic DNA arrangement. But I'm not aware of any papers on that.
I'll tell you of a paper, and it was written by Change Tan and Jeff Tompkins. I really wish they had tried to get in a secular journal because it was so good. Mm-hmm. It was really so good because she took Change Tan, professor molecular cell biology, until she got dismissed from
tenure.
Oh gosh, Mizzou, University of Missouri, sad. Yeah. And I think I'm okay sharing her personal conversion story. I asked her, no, she did this publicly on my channel.
Yeah, I thought she did, yeah.
Yeah, I asked her, how did you become a creationist? And I don't quite know the sequence of how, when she became a Christian and when she became a creationist. She came from communist China and, the non-Christian viewpoint. And so that says it all. She came to the United States and she was a physical organic chemist. Physical chemistry is hard. Organic chemistry is hard. She's a physical organic chemist. 2 hard fields. And she got her PhD at an Ivy League school, University of Pennsylvania, her postdoc at Harvard. Somewhere along the way, she met eventual Nobel Prize winner in either physiology or medicine or chemistry. It was Smith, George Smith. I know that's such a typical name. And he said, Dr. Tan, I think you should be teaching molecular biology. And she had all the qualifications as far as the chemistry, and her postdoc was kind of in cells. It's different than when you actually have to teach a class. So she learned all this. Mm-hmm. As she studied the difference between just the molecular biology side, which is transcription, translation, and everything just kind of around that. Between eukaryotes and prokaryotes, she said, no, this couldn't have evolved. And she covers all of this. She covers a lot of this, and I'm just like, yeah, how can anyone think this evolves naturally? This isn't, at least not by gradual, small, incremental steps. And she said that's the moment she became a creationist. I know how the conversation came up. I said, how did you decide to write The Stairway of Life? She said, it actually began when I was having to teach molecular biology, and I saw the different translation apparatus between prokaryotes and eukaryotes. And then even then, between archaea, bacteria, and eukaryotes, she was just like, wow. Not necessarily transcription translate. Well, yes, there is. There are funny things like with the helicases that don't suggest common ancestry. You'd have to invoke miracles. And she said at that point she became a creationist, and she started examining. She said, let's go a little bit further, a little bit more elementary. And that was the beginning of the Stairway to Life. So that's discontinuity systematics again. Yeah, I wish, I'm not into baraminology, but I am into discontinuity systematics. I love this stuff. So sorry, I just got excited just reading this Wikipedia article. Holy smokes.
I can pick up on the introduction if you'd like me to.
Yeah, can I this was just a little too interesting here.
That's fine.
So most vertebrate telomeric DNA consists of long TTA GGG repeats of variable length, often around 3 to 20 kilobytes. In vertebrates, each chromosome, subtelomeres are segments of DNA between telomeric caps and chromatin. In vertebrates, each chromosome has 2 subtelomeres adjacent to the long TTA-GGG repeats. Subtelomeres are considered to be the most distal, farthest from the centromere region of unique DNA on a chromosome, and they are unusually dynamic and variable mosaics of multichromosomal blocks of sequence. And I think there are different implementations of the telomeric repeats because the vertebrate telomeric repeat is different than others, which is another discontinuity. Oh, I love discontinuity systematics. Anyway, so let us continue. I'm sorry, Emory. I got so excited reading all this stuff.
It's fine. So introduction section. Tera transcripts are nuclear long non-coding RNAs that are transcribed from the subtelomere towards the telomere. Okay, so they're reading toward the end of the chromosome. They are transcribed by RNA polymerase II. That's up your alley, Sal. Giving rise to transcripts that contain UUAGGG repeats, being the presence of this repeat their main features.
Oh, I need to point something out. So if you transcribe, let's go back to the Wikipedia. I'm sorry. I'm getting too, I'm just giddy with excitement here. If we look, the transcription of a telomeric repeat would be UUAGGG. UUAGGG. And so here it is saying, where did it say it? UUAGGG. So that is the same as the telomeric repeat right there. Isn't that awesome?
It is interesting, yeah.
And I got to point out, again, for discontinuity systematics, you'd have to account for that.
Yeah, you would need to find a way to account for those changes in a evolutionary model.
Oh, they'll just do a phylogenetic reconstruction. We know how they do their thing. It happened. Okay.
Let's see.
Okay, here it is. I'm sorry. That
Fine.
transcript that contained UAGGT repeats.
Mm-hmm. They are.
Oh my goodness! By the way, can we say hi to all the nerds that joined us?
Yeah.
We have Dr. Joel, we have Redeem Team, Mark O'Connell, and Ray Giordano. How about that? Oh my goodness! Some guys are into some serious stuff here. Whoa!
Yeah, you need to be a real big nerd to be paying attention to this. All right. Finishing paragraph 1. They are also heterogeneous in size, 0.2 to 10 kilobases in humans and mice, as indicated by the smear detected in Terra northern blots. On the other hand, detection of Terra by RNA fluorescence in situ hybridization, FISH, renders a clear spotted pattern in the nucleus, and the number of spots varies in different cell types. Approximately 30% of these spots co-localize with telomeres. Lack of Terra's sub-telomeric sequence information has been an important shortcoming to understand the role of Terra, as most of the functional studies published to date are based on the use of the UUAGGG tract to detect Terra. Thus, Terra have been implicated in telomere protection, heterochromatin formation, telomere replication by sequestering RNPA1 from telomeres to allow RPA to POT1 switching, a process that is altered in alternative lengthening of telomeres cells lacking ATRX, and in telomere elongation by homologous recombination through the formation of DNA-Terra hybrids. That's a lot of stuff, and I'm not sure I understand all.
Okay, so guys, the guys who came in late, why is Sal just so thrilled? Because the evolutionists are proven wrong yet again. And they were just saying, this is all junk. It's all junk. There's no designer. And science is just whipping them to death now. It's embarrassing. I mean, and I just commented earlier, I said, oh, I got a private message from Emory here. I'm sorry. Can I share it? I mean, you're
Sal, read it before you do that. Read it before you do that.
Okay. Nature Communications, all the peer review that goes on. Let me see who the guys who wrote this. All right. Just as a matter that I've noticed, a lot of good Nature articles have to come from people outside the United States.
Mm-hmm.
And guess who the peer reviewers are probably going to be? Also people outside the United States, because
most likely,
yeah. All right. So there is some concern about typos. It just happens. Okay.
Yeah, I know. I mean, I want to be open and above board with it, because as I'm reading it, I'm catching typos, and I'm not an editor. And that now it seems like most of the people who were writers for this were not native English speakers. All the last names are Hispanic.
Yeah.
So I'm not trying to rip these people at all. But I am concerned about the review process for this article, because if this article went through peer review correctly, or went through editing after it went through peer review, that should have been cleaned up. And okay. I'm through 2 paragraphs, and I've got 3 grammar mistakes already.
And I didn't see it. Okay, so all right. I mean,
now, and I'm not saying, by the way, I just want to point out, I am not saying that the results in this article are wrong just because they have a few typos.
Yeah.
Okay, that's not the claim, but I am a little bit concerned that Nature Communications did not pick up on that.
Right. Okay, can I express a bigger concern?
Sure.
I am concerned that the scientific enterprise has let tons of articles and journals things published on evolutionary biology. I think that's the bigger failure. Okay.
I'd agree with that.
If we're going to let garbage like that get through scientific peer review, hey, I'm going to let grammar mistakes get by. Just blowing off a little steam here. The important thing is this is subject to replication. And I do think their methods were important because they did a knockout experiment and it in the subtelomeres and it caused damage. That's good enough for me. So guys that joined us late, and I'm sorry to have brought that up, but I felt kind of honor bound to at least mention it. If you're concerned that I felt I should say something too. So caveat with everything. All right. But this is, I think it indicates good science. So guys that joined us late, why am I so excited? Evolutionary biologists were just screaming at ENCODE saying this is all functional. And, here's a section of about 2% of the DNA that even I thought might not have any use if it ends up transcribing to RNA. Obviously, the DNA had function, but I said there's no reason to transcribe it. I said maybe that's a spurious I honestly thought that could be a spurious transcription, but you never know. That people would have been jumping all that. See, it's like, that's bad design. Look at all that spurious transcription that doesn't do anything. And it's like, nope, I think that was premature because we're finding a lot about the biophysical properties of RNA. And, that's subject to another talk. That's why I was excited, because anytime we have a chance to take it to the evolutionary biologist, I'm just thrilled. I know that's not very sporting. That seems kind of mean-spirited, but hey, that's just me. So that's just to catch people up.
Yep.
This is great stuff. So thanks, Emory. So we are at the DNA Terra hybrids.
Yeah. So DNA Terra hybrids. Let me find the paper again.
Oh, can I go to my ad blocker screen? I'm sorry. Yeah. There, we don't have those obnoxious ads.
Yeah.
Okay, there it is. Terras are also proposed. Okay.
All right. So, let's see, where was I? Oh, yeah, that was okay. Terras are also proposed to bind the telomerase core components, TERC and TERT, but their role in telomerase function is unclear. Sal, do you want to explain what telomerase is?
I think they call that the immortalizing enzyme. So let me just say, oh my goodness, I got to tell you, I was at the ENCODE conference, 2000 users conference, and I got to be beside a researcher on telomerase. And I said, now they've said that if we can reprogram the telomeres with telomerase, we live forever. He said, no, that's not true. Mice have long telomeres. They've got, yet they die. But I was still fascinated. So immortalizing enzyme. Ah, yes. All right. It is called the immortalizing enzyme. I just wanted to make sure I got this right. So here we go. The immortalizing enzyme. In the early 1500s, according to legend, Spanish explorer Ponce de Leon traversed Florida looking for the fountain of youth. He never found it. 500 years later, scientists are still searching for it. They haven't found it either, but they might be getting close. No, they're not. And so there is this thing, a certain anti-aging enzyme has captured the attention of the scientific community. Telomerase is the name of this immortal enzyme. Okay, so that's all I'm going to say on that, okay? That was just kind of interesting. So it generates the telomeres, that TTAGGG pattern. And so this is actually one part of DNA that's actually not replicated in the usual manner. This is, as I understand it, it's the telomerase that will actually construct it. So most of the DNA is replicated, but then you have a special enzyme that will actually create it. Now, the mice have a special kind of telomerase that keep their telomeres long. Now, our aging process is correlated with the loss of the telomere lengths in our cells. And it seems to be it is correlated with cancers and aging. And there's one thing. It also deals with what they call the Hayflick limit. Because once it reaches that Hayflick limit, the telomeres are too short, the cell can't reproduce anymore unless something special happens. Something like that. Dr. Joel said, okay, so we'll just stay on topic. So here it says, TERA are proposed to bind to the telomerase core components TERK and TERT. So TERK and TERT would be the domains inside the telomerase core. I'm just presuming this. Okay, so that means a protein usually has component parts. All right, so that's about all I know, and we can go on now. By the way, more discontinuity systematics right there, baby.
So I'm actually going to answer a point raised by Dr. Duff, because Dr. Duff is pointing out that in his video, and I think I'm reading this correctly, if you generated 5,000 random NT, and I'm assuming that's nucleotide triplets. I'm not entirely sure what NT stands for. Sal, do you know that? Or
nucleotides. Yeah.
It's just nucleotides. Okay. And inserted them into a genome, would that DNA be transcribed? The answer is that it most likely would be. Yeah, I'm not going to disagree with that, but transcription of DNA is not necessarily a sign that the DNA is functional. I'll grant you that. But the problem is we find functions for the vast majority of non-coding RNA. We found functions for it. I mean, and I did, just as a 5-second Google Scholar search, I typed in function of non-coding RNA, and I got thousands of hits.
Yeah.
So I don't see that objection really holds any water whatsoever.
Yes. Against the design argument or that evolutionary biology is correct. I also think, as having worked on the genetic entropy project, I think there is a lot of junk DNA. We don't know what fraction there is. I personally think we've lost a lot. What do I mean by a lot? I don't know. I mean, I don't think it's, John Sanford has quoted Kondrashov, who says we've lost hundreds of genes. Maybe it could be 1,000 per person. I mean, that's like 5%. Yeah. I think we've suffered some genomic damage. I don't think the human race could tolerate much more than 20-30% like bacteria could. So
no, I don't think so.
Yeah. So I will say there's junk, and we do know that bacteria can transcribe stuff that they don't even use. That's how come we have all these medical things like bacterial made human insulin that we use for medical, to treat diabetic patients. So absolutely you'll transcribe stuff that is junk to the organism. Mm-hmm. So anyway, thank you for responding, and thank you for your comment, Dr. Joel. So,
all right. So I'll pick up where I left off, which was role in telomerase activity. Okay. On one hand,
TERA can inhibit telomerase activity in vitro.
Yes. On one hand, Tera can inhibit telomerase activity in vitro. On the other hand, in yeast, teras are induced at short telomeres and form Tera-Turk RNA clusters that co-localize with the telomeres of origin during S phase, suggesting that Tera may play a role in the spatial organization of telomerase activity at telomeres. Now, I'm going to raise a small red flag here, because we know from now, if the Wikipedia article is correct, we know that telomeres in vertebrates operate differently than telomeres in invertebrates. So the yeast example may not be relevant because they're talking about human telomeric RNA in this article. However, it is conceivable that it is. We don't have that information. So small red flag. It's not a major deal, but it's just something worth pointing out.
Oh, Janet's here. Janet's here. Can we say a prayer for Janet? Let me just say a prayer. Heavenly Father, we offer this widow and disabled lady into your hands. Please, all Christians, join us in praying a prayer for Janet Duttridge. Lord, you've said true religion is to attend to widows and orphans in their distress, and we do that tonight. Glory and honor to you, our Lord and Savior, Jesus Christ, and please be with this stream. In Jesus' name, amen. It's a pleasure to serve you, Janet. God bless you. Thank you for that caveat. See, I love your skepticism. This is just wonderful, and that's okay.
Yeah, I mean, I have learned from experience and writing my own thesis for my master's that it is wise to be very skeptical of things that you are told in the peer-reviewed literature, because people have a human nature, and because they have a human nature, they will find papers to support points, even if they don't support the point, or we can't be sure if they support the point, because they need to make that point for their paper that they're submitting to be accepted or whatever. And this may, and by the way, I do not want to blame the authors here. The reviewers may have said, well, there's this paper over in yeast here that you should cite. And if your reviewers tell you need to cite a paper, you pretty much need to cite that paper or you're not getting published. So I'm not necessarily blaming the authors here. They may have, some reviewer may have reviewed this and said, this paper in yeast would be a good fit here. You should add it. And they went, okay, fine, we'll add it so we get published. Okay, I'm just pointing that out as a possibility.
Yes. Oh, by the way, it says, on the other hand, in yeast.
Yeah.
I think they had put the proper qualifier there.
Yeah,
people that are in the know.
The qualifier is that the Tera, the Tera is doing different things in different organisms. So they're pointing out that there is potentially a contradiction there, which I appreciate them doing that. But I do just want to point out that the yeast stuff that they just mentioned and that they're going to talk about in the next paragraph might not be relevant. Just maybe. I'm saying maybe. Right. I don't know. We don't know.
And you know what? That would be fine, because if they are different, that means more discontinuities.
I thought you'd like that.
It means God did it, not an evolutionary process. So whichever way this plays out, we're going to have fun. Praise God.
All right. So moving down to the next section, I think this is where they start getting into some of the work that they did. In yeast, several Tera transcription start sites have been identified at different chromosome ends. In the mouse, using a genome-wide RNA sequence approach, we recently identified that transcripts showing bona fide Tera features mainly arise from the chromosome 18 subtelomere, and to a lesser extent from the subtelomere of chromosome 9. Further, we found that chromosome 18 Tera associate in trans with the remaining telomeres and are important for telomere protection.
Whoa, 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 whoa! Bravo!
Yeah, that's mice, by the way.
Well, still, this is trans chromosomal regulation.
Yeah.
This is awesome.
Okay, so let me just raise a point here. Bacteria have 1 or at the most 2 chromosomes. I don't know how many mice have, but humans have 46. Chimps have 48. There are some organisms with several hundred. Please provide me a paper that explains how trans-chromosomal regulation of telomeres evolved from a single chromosomal ancestor without a phylogenetic reconstruction. Because I can make one of those, and I can make it say any bloody thing I want it to say.
And even if you assume common descent with a phylogenetic reconstruction doesn't describe the feasibility in terms of probabilities.
No, all it provides you is a potential pathway. It's a hypothesis and nothing more.
Yeah.
It's not data. So,
anyway. Okay, now I got to pause a little bit, and I'm embarrassed to say this. So there's an S phase in a cell and then a G phase. It mentioned it earlier in the article with the yeast.
Yes.
What is the S phase again?
Okay, so the cell goes through 3 phases. S phase is when DNA is replicated.
Okay, thank you.
Which it would make sense that that telomerase would be involved in that, because during replication would be when the telomeres are most likely to be shortened. That the Tera stuff would be involved in that, because during replication is where with the telomeres would most likely be shortened when you're replicating, because you could easily leave off a bit on the end.
All right.
Does that make sense?
Okay, this is okay. Let's. Well,
yeah, that would help. That would help.
DNA replication. So we have G1 phase. You have the G0, G1, S phase, G2.
So G1 is you've gotten a new cell. 2 cells have divided. You now have a new cell. The cell has to grow and mature. Then it goes into the DNA replication phase, which is the S phase. That's where you make a second copy of your DNA. Then you go into the G2 phase, which is where you start preparing for mitosis or meiosis, depending on which type of cell we are. And then you go through mitosis, and you come out with 2 cells in the G1 phase again.
Holy smokes!
That is a very, 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 very simplified version of the cell cycle.
Oh my goodness! And there's interface. Okay.
Yeah.
So I thoroughly embarrassed my
interface is everything except mitosis. Everything except the mitosis section. So interface covers G1, S, and G2.
Gotcha. Oh man, I'm so glad we're reviewing this. Okay, I tell you what, I'm a fast learner, but I'm a fast forgetter too. The only class I ever got 100 through the whole class was in cell biology. And you could see how quickly, I think after 2 weeks, it was all gone. So this is a great refresher. I'm sorry to say that's just, that's why I was able to get through school, is because I was a fast learner. But once the exam was over, it's like, okay, I only have so much random access memory. I got to clear it out to make room for the next. So, okay, thank you. So we were here with trans-chromosomal regulation.
Yep. Tera loci in human cells have been more elusive. First, a putative Tera promoter was proposed to consist of a 61, 29, 37 repeat, a conserved region that contains 3 different repetitive DNA tracks of 61, 29, and 37 base pairs, which is present at 20 different chromosomes. However, transcript sizes and their regulation were not identical to those of Tera, suggesting that they may constitute a fraction of Terra molecules. More recently, a similar RNA-seq approach that used by us in mice. Okay, that makes more sense. To that used by us in mice, identified 10 distinct human chromosome ends where puntative Terra transcription started as far away as 5 to 10 kilobases away from the telomere, as well as 8 additional chromosome ends where transcription started in close proximity to the telomere. The authors, however, did not address whether these transcripts showed Terra features defined as the presence of UUAGGG repeats within their sequence, their heterogeneity in size, and the nuclear spotted pattern in which some spots co-localize with the telomere. So apparently another paper had found a bunch of potential Terra locations, but did not test those locations. And so they appear to be building on that. Go ahead.
Apologies first to the viewers. I messed up sharing my screen for what Emory was reading. I got to tell you something that just leaps out at me. This means that this regulatory sequence has the TTAGGG repeat that transcribes the UUAGGG repeat. I think this does not look like random insertions to me.
Yeah, they don't look like random insertions to me either.
I mean, this looks pretty, like designed and crafted.
Yeah.
All right.
That's what it looks like to me, too. And it's going to look more like that by the time we're done.
This is also saying, okay, guys, just got to mention this. We're finding that repetitive elements have biophysical significance. The RNA will have biophysical properties because of a repeat. And I saw, as anyone that does engineering and is fascinated by material science, and repeat is the order of the day if you want to make a homogeneous material that has physical properties. That's just the way it goes.
Mm-hmm.
So the fact it never translated to anything, I'm like, and the evolutionary biologists are saying this is junk. I said, no, I think it could have biophysical uses. Exactly what we're finding. Oh, well.
Yep. I would. Yep. So
take that, Larry Moran. I'm sorry. Couldn't help myself. Go ahead. Thank you so much. Thank you so much for reading this.
That's fine. So here we study whether these human transcripts have the above mentioned Terra features, with the final goal of functionally deleting them to unveil the role of Terra. We found that out of the 18 proposed Terra loci in humans, only transcripts arising from 20q and XP loci have Terra features. We then used CRISPR-Cas9 technology to genetically delete these 2 potential Terra loci in humans to study whether they are indeed the origin of Terra. Only deletion of the 20q locus caused a dramatic decrease in Terra levels, while deletion of the XP locus did not result in decreased Terra levels. Importantly, the deletion of the 20q Terra locus leads to telomere shortening and telomere uncapping, as indicated by increased telomere damage foci, or TIFs. These unprecedented findings demonstrate that Terra transcripts are essential for the maintenance of a functional telomere cap.
TIF, what is a TIF?
Telomere damaged foci.
Oh, okay.
It might be TLF. I'm not sure. But
all right. Well, we'll go on here. Yeah. Any comments? I'm loving this, man.
I'm looking at that, and this, Sal, I don't know if you realize how long this paper is, but I'm looking at that and going, oh boy. it looks like there's only one place, roughly, where we make all of the, I mean, provided they did not review all possible places, just some places, 18 proposed terra loci. But of those 18, only one of them, on chromosome 20, actually controls the breakdown of telomeres for all other chromosomes.
Okay, let me point something out.
Sure.
We only do experiments on a very limited number of cell types.
Mm-hmm.
We have no idea how easily we can generalize what happens in one cell type to another.
Yes.
I'm not going to be surprised if we're like, oh, this doesn't work for this cell type.
Mm-hmm. Yes.
So, I mean, God's showing his great power and wisdom here. So now, granted, the paper is long. Let me just do a quick look to see how much of this is the citation part.
Not that much.
Oh my goodness. I'm open to suggestions here, but I'm loving this so far.
I mean, it will be very interesting. I just don't know. I don't think there's any way we get through this tonight.
Well, you know what? Maybe we can skip the methods if we just get to results and discussion. I got to
Result is next.
Yeah, I got to tell you something. In general, I'm really glad when we see researchers go through the trouble to have materials and methods. That means other people can duplicate the results and inspect it.
Yes.
So I've been writing I wrote a paper with Dr. Sanford. The materials and methods section was brutal, but I'm going to make sure anyone can replicate what we did.
Yes.
So
I agree. So I think we should, Sal, the results section is really long.
Okay.
The discussion section is very short, and the methods are at the end. So we'll just completely skip those. I think we should focus on the figures because this is video media. So let's maybe use the figures that are available to us in the results section because we will not finish the results section tonight.
Okay. You tell you what, we'll just skip it altogether.
Okay.
Because once right.
Okay.
I got to tell you,
I will go through and read the data later and just see what I think about it. But I want to we'll go down to the discussion for sake of time because otherwise we'll be here till next year.
Okay, so just briefly, I thought I saw that the green identifies the loci of interest. I mean, this is what they have to do to figure out where things are. Yeah. I mean, this is tough research, guys.
Yeah,
at the nanomolecular level. I could tell you it is comforting to see their methods here. This is what I call really good,
Yeah. This makes me feel better about the typos, looking at this stuff and going, this looks like they actually did significant work here.
Yeah, which is more than I could say about evolutionary biology papers. I'm sorry.
Okay, so this paper, if you tried to print it, would probably be what, 15, 20 pages, Sal?
Yeah.
Most evolutionary biology papers struggle to hit 10.
Oh, okay. So they're showing. Have you ever done blots, northern and?
I've done a few. I've done some in my time. It's been a while, but I've done some.
Okay, so guys, this is how sometimes we're able to probe the molecular details. We create all these blots, and it's just so much of this is by inference because we cannot see what is there except indirectly.
Yeah.
We have to reconstruct the way the molecules attach to each other. And there's this northern blot, southern blot, western and eastern blot. It all began with a guy named Southern. His last name really was Southern. Mm-hmm. And people started making fun of, they call it the southern blot. So someone came up with the northern blot. That used to throw me off. So, okay, guys, I'm just scrolling through how this is agony to figure this out. So if for nothing else, you can appreciate real scientific work here.
Yeah, this is good work here from the look of it. I will have to read the data and read the methods later to get a firm handle on it. But just from the feel of it, it looks like they put in quite a bit of work.
This is, yeah. I'm glad they're showing all their panels. I could tell by the quality, they were very serious about doing this. God bless them. Okay, so here we go, the discussion.
All right, so the discussion is kind of similar to what we've already read in some places, but repetition is the mother of learning or something. Here we identified a major locus for human Tera transcription at the subtelomere of chromosome 20q. The genetic deletion of this locus allowed us to demonstrate for the first time in any organism the essential role of Tera in the maintenance of functional telomeres. That's big stuff right there, by the way.
Yep. And by the way, if I may interject, reading the earlier parts, they are not unique in this. There was other work done on this. So they're building on other people's work. This is not an isolated case of functionality. And I got to tell you, way back in 2015, when I was at the ENCODE conference, I didn't, and even like a couple years thereafter, I didn't know about this work. So that would mean even while I was at the ENCODE conference, while Dan Grauer, Larry Moran, and others like the editor of Cell Reports, Steve Matheson, were just on the junk DNA bandwagon. There is lots of literature out there that I just was not acquainted with. So anyway, go on.
Yeah. And the literature just keeps coming in that direction. Junk DNA, outside of evolutionary biology, junk DNA is sort of dropping backward toward it's kind of falling backwards a bit. Yeah. So anyways, moving on. In a previous report, 18 different human subtelomeres were proposed to contain Tera loci, although the authors did not study whether these transcripts had indeed Tera features. Here we found that only 2 of the 18 loci have Tera features, in particular those corresponding to 20q and XP subtelomeres. Furthermore, on genetic deletion of these loci using CRISPR-Cas9, only the 20q locus was confirmed to be a bona fide Tera locus, since it resulted in almost complete abrogation of Tera expression. By the way, the fact that deleting that one locus resulted in almost complete deletion of Tera expression means that most, if not all, of the Tera that is in a cell comes from that locus.
Yep.
And what that means is, since humans have 23 pairs of chromosomes, one locus is regulating the telomeres on 23 pairs of chromosomes. Okay? Just think about that for a second and then ask yourself the question, how did that evolve? Right? Anyway, moving on. And by the way, for any Evos who want to give me an answer to that question, send me papers. I'll read them. Moving on. Failure to decrease Tera levels on XP deletion may be explained by the impossibility to delete the entire locus due to the highly repetitive nature of this region, or may indicate that this is not a bona fide Tera locus. Okay, I like that. They're hedging a little bit. That's good. Alternatively, our finding that 20q Teras are increased on deletion of the XP locus may also explain the fact that XP deletion did not result in decreased Tera levels. Oh, that's interesting. That looks like 20q Tera is upregulated when the XP Tera is eliminated. Is that what you're seeing?
Yes, the XP is an inhibitor.
Or the cell is able to sense its loss and upregulates the one on the 20 chromosome.
Yeah.
That's
Wow.
really interesting. I'd like to know how the cell does that.
Yeah.
That's really interesting. Now I'm gonna make a I'm gonna make a wager that's probably foolish, but I'm gonna make it anyway. I will wager that when they find these Terra-specific regions in other organisms, that the Terra-specific regions regulating the telomeres in those organisms will not be on those 2 chromosomes.
I think you've got a good wager there, and just visually, if I may, if you'll indulge me here.
Sure, go ahead.
Let's look at the difference between mice and humans. There is one diagram here because you could, the trans chromosomal regulation will be different, and we just talked about trans. All right, so this one diagram really. All right, so you see, like in a mouse, if you take all the pieces of the chromosome, and let's say we evolved from a mouse-like creature, look at how all the pieces of the mouse chromosome have to map into the human chromosome. So what was that, like 18 here?
Yeah.
Yeah. Yeah, baby. Discontinuity systematics, baby.
Oh.
God bless you, Walter Remine. If only we could hang out and honor your work. Biotic message all the way.
Mm-hmm. Yep. One of the best books I've read. I need to do a review of it one of these days. Anyways, picking back up where we left off. In any case, the fact that deletion of the 20q locus results in dramatically reduced Terra levels demonstrates that this locus is a major Terra locus in human cells. Interestingly, the finding of 1 or 2 at most Terra locus in human cells resembles the situation recently described by us in the case of a mouse Terra. In particular, by using a similar approach to that used in humans, RNA-seq following UUAG GG transcript enrichment, we identified mouse transcripts arising from 13 different chromosome ends, but only those transcribed from the subtelomere of chromosome 18 and 9 were confirmed to be bona fide Terra transcripts, being the chromosome 18 locus responsible for the majority of Terra transcripts. I hadn't read that, folks. I should have bet the house on it.
So what was your bet again?
So basically, I said that when they find the subtelomere regions that regulate the ends of chromosomes, that they would be on different chromosomes than the human ones were.
There you go.
And
Number 9.
I had not read that paragraph yet.
Should have bet the house, baby.
I should have. Oh my! I thought I was actually taking a risk. Okay. I'm not a prophet. All right. But yes, so 18 and 9 compared to humans 20q and x. So thus, in both human and mouse, Terras are transcribed from 1 or 2 subtelomeres, a finding that was not anticipated since it was widely accepted that Terra were arising from all chromosome ends. It remains to be deciphered whether Terra transcripts play different roles depending on the locus of origin.
Whoa. So
that's a heck of an admission.
I mean, this means that chromosome organization it's pretty important for the regulation of all the other chromosomes. Wow.
Yeah, that's
I mean, you will not have all those chromosomes if you don't have at least one particular one.
yeah, that's whoo.
Oh,
Okay.
more discontinuities. Take that, evolutionary biologists.
Mm-hmm. Yep. Discontinuities galore. Importantly, here we describe the first genetic deletion of Terra performed in any organism. They've said that about 4 times now. Findings are striking as they clearly demonstrate an essential role of Terra in cellular viability as well as in telomere maintenance and telomere capping. In particular, the impossibility to expand most of the homozygous clones for the 20q deletion in 3 independent human cell lines, HeLa cells, HCT116, and U2OS, suggests an essential role of Terra for cell survival in the majority of cell lines studied here. Only the U2OS cell line, which expresses higher basal cells of Terra, could bear the 20q deletion, suggesting that the cell needs to maintain a minimum amount of Terra levels to survive. Well, that's interesting because I don't know. I know the HeLa cell lines were from a woman who had cancer. They're cancer cells,
Right.
I don't know the other 2 cell lines. And they're, I hate to say that, they're probably aborted fetal tissue, to be perfectly honest with you.
Yeah.
But I don't know what body parts, and I hate, this is depressing, but I don't know what ghoulish method they use. So I don't know, like, there may be different cell types here. And if there are different cell types here, then Sal's prediction about different cell types having different expression amounts of Terra might be also true. Okay, so Mark came in late. Let me refresh everyone on what Terra means. Terra is long non-coding RNAs that are transcribed from telomeres or subtelomeres.
Okay. So just since I'm actually kind of new to all this, Terra is an abbreviation for telomeric repeat containing RNA. Terra. Terra is RNA that is transcribed from telomeres, the repeating 6 nucleotide sequence that caps the end of chromosomes. Terra functions with shelterin to inhibit telomere lengthening by the enzyme telomerase. Terras are essential for telomere length and maintenance. At least 4 factors contribute to telomere maintenance: telomerase, shelterin, Terra, and the CST complex. Terra can also regulate telomere length by increasing euchromatin formation. On the other hand, nonsense-mediated decay factor enrichment of telomeres may exist to prevent Terra inhibition and telomerase. Terra levels may vary during cell cycle, decreasing during S phase and increasing transition from G2 phase to G1 phase. All this to say, the prediction this was junk is junk. That was nonsense. That's what you get from evolutionary biology. You listen to them, you'll get it wrong. That's the bottom line. That's what we feel anyway. Kind of another, wound, another jab at the evolutionary biologists. They're saying this is all junk. And as you hear this complex description of Terra doing it, even if you don't understand all the terms, you can get the sense this really does something, and you'll be dead without this stuff, which the evolutionary biologist said you don't need. Evolutionary biologist, wrong again. No surprise.
Yeah, I've come to the conclusion that 9 times out of 10, when an evolutionary biologist opens his mouth, he's wrong. Let's continue with the discussion section. Importantly, we found a dramatic loss of telomeric sequences in U2OS cells on the deletion of the 20q tera locus, indicating that tera is essential for telomere maintenance. This dramatic telomere shortening was accompanied by telomere uncapping, as indicated by increased telomeric damage and increased telomeric fusions. Interestingly, U2OS cells are known to maintain telomeres by homologous recombination, a process in which tera has been suggested to participate. In line with this, we found detectable but significantly decreased telomere recombination frequencies, as indicated by TSCE frequency. No clue what that is. Together, these results indicate an essential role for tera in cellular viability and in the maintenance of telomeres, even in cells that maintain telomeres by recombination. Interestingly, deletions and amplifications of the 20q subtelomere have been found in patients with different types of hematopoietic malignancies and with mental retardation.
Wow. Wow.
Oh, hello.
Hello.
That's interesting. So I'm just looking at their references here. The diseases associated here are Jacobson syndrome, oh my goodness, I can't pronounce this, myelocytic disorder, mental retardation, and whatever hematopoietic malignancies means. I'm not entirely sure what that is. I'm not a doctor. But none of that stuff sounds particularly good.
No.
It's rather bad, honestly. And this is the stuff that that is the problem with if you don't have these telomere non-coding RNAs. And non-coding RNAs are just junk, huh?
Yeah.
Okay.
And you know what? Why don't you, if you could read the last sentence, and we can just kind of Sure.
Although some of these deletions also include coding genes, it is tempting to speculate a specific role of the 20q tera on these malignancies. Finally, the finding of human 20q tera locus described here warrants further studies to analyze of a possible association of 20q tera to these and other human diseases. So in other words, we want to study this more, but it looks promising as a potential cause for human diseases.
Yes. And by the way, just to honor their hard work, I mean, look, this is how hard they had to work to it's painful just even reading this, much less executing the experiments and then reading all these citations. This is why science is so hard. And, you know, hats off to them. And I would like to salute them.
Just as an experiment, I just quickly went to look to say, okay, how long would this be if I printed it? It's 37 pages. And there's only like 20 some references because this is an experimental paper. So there's not as many references, only like 30 references or something. So they did mighty amounts of work on this. Or they did mighty amounts of fraud, one of the two, but I suspect they did mighty amounts of work.
Yeah, that's the thing is when you have an experiment that can be replicated and other people can work on it. And the other thing is in the medical science community, if you screw up and there's fraud and this is earlier in your career, you're done.
Yeah, you're toast. You're over.
Yeah. But if you provide something that will advance disease, understanding and curing and treatment of diseases, that starts to establish a reputation. And the other thing, honestly, there's a lot of motivation to be honest in this and to be careful because a lot of people have sick relatives and you take this as sacred work, whereas making a phylogenetic tree, what's at stake? Honestly, it's just trying to get part of the club. let me see. Let me make that phylogenetic tree kind of agree with the editor what he wants to see.
Yeah.
I got to say, John Avice, no intelligent design. Okay, I have this book, and it's. I want to show you and contrast that paper to this particular book by John Avice, Inside the Human Genome, A Case for Non-Intelligent Design. And he goes on and on. He said, there's no intelligent designer. Look at all these repeats. That's so stupid. Duh. And we have just shown what happens when you don't have those repeats in the case of the telomeres. This is bad news, and not just the repeats at the DNA, but at the RNA level. It's really important. Mm-hmm. And we don't completely understand why, but I'm like, if you're a materials engineer, it's like, I'm not surprised when you have materials that are homogeneous. It's kind of nice,
Mm-hmm. Yep.
So, and I bet you there's going to be a discontinuity. I'm not going to bet the house on this, but there's going to be an interesting discontinuity in non-vertebrate telomeres because they have different spelling, and who knows how that is going to play out. It's not as trivial. This whole thing about coevolution, it's like we made a phylogenetic diagram. Look, it makes sense. It's like if you tweak something here, you got to tweak all the proteins that bind to that location. All right, this is not trivial. I'm just like, guys, do you not even think? Of course, that's a rhetorical question. I know the answer to that, but it's hard to do coevolution when you have to get simultaneous changes in so many other systems to make a change here.
Yeah. Yeah, so I was reading a book recently by oh goodness, I forget the name of the author, but his argument effectively was that it was very easy to get in a large enough population to get a beneficial mutation. And I thought, well, okay, you can get one mutation. Okay, maybe you get a particular mutation, but that's not your problem. That's not your problem. You've got to co-evolve that mutation with every other mutation in every other gene simultaneously, and you got to spread it through a giant population, which, given that it starts with a frequency of basically 0, is very, very unlikely.
Yep. I tried to get it through Evo grad students' head. You can't do this. And I specifically cited zinc fingers, and he just did phylogenetic diagrams and said I just like, dude, you have no comprehension of how protein biology actually works. You may, but you're kind of pathologically blinded by your own profession. I'm sorry. It's pathetic.
Yeah.
And that's why I was fed up with Swami Das's website, because these guys were just numbskulls. You could lead the horse to water or whatever they say.
Yeah, there's only so much. I'm sure they're very intelligent people, but they're willfully ignorant. And I hate to borrow that phrase, but they're willfully ignorant, because a lot of them they bought into so many false assumptions that they can't see past.
Okay, I'm going to be harsh here, and I'm just sorry to put this out, but I mean, we can come up with our explanations. But Jordan Peterson and Abigail Schreier pointed out a phenomenon where being part of the tribe, we see this in anorexic girls in particular. They're persuaded that they are fat when they're skinny thin, on the verge of dying. Really tragic situation.
Mm-hmm. Yep.
It can be reinforced if you're in a community with other anorexics, so they try to separate them so that they don't reinforce each other. And Abigail Schreier pointed out, she said, this is an illustration of what they call psychic pandemics, where being part of the tribe is more important. You're willing to deny reality just so you could be part of the tribe.
Mm-hmm.
So
that is where academics live.
That's academia. And so they're like anorexic girls. All right, I know that part. Go ahead.
I'll even explain why this happens, and I'll use Dr. Dan, because Dr. Dan actually has said this to me. I don't know if he remembers it, but we had this, we discussed this, where where we were talking about imposter syndrome. We all feel like we're the dumbest person in the room. And I asked him, does that ever go away? And he said no. So effectively what's going on with a lot of academics is they've got imposter syndrome. They think they're too dumb to be there, and that they're the only ones who don't belong in the room. And they think everybody else around them super smart because they're reading everybody else's papers and going, oh my goodness, this guy's done something really great over here. I don't know what's going on I have no clue what's going on in my lab, but this guy over here, he's got it all together. And they're terrified, absolutely terrified of being thought that they're stupid, because they'll lose all their prestige and their positions and everything else if they say something that's stupid. So they repeat the same lines that everybody else says because they think that somebody else has the answer. They don't understand it, but they're sure somebody else does. And so they all echo the same talking points, because even if they don't necessarily, or if they've got questions about the talking points, they think, because they've got imposter syndrome, that I don't belong here. If I say something stupid, I might lose my job. So I got to be really careful. I don't want to say something dumb. These other people, they're really smart, and they've got this. They say it's this way, so it's got to be that way. Not knowing that the people making the comment are thinking the exact same thing that they're thinking. Oh, that other guy over there, man, his lab is so well ordered. He's got everything. He's right on top of everything. He's got so many great papers. And my labs are falling apart. My grad student just blew up the chemistry department. I mean, what? So they're all thinking through the same mindset. Because in academia, it's very insular. They have this. There's a feeling of you're superior to the mob, to the general run of population, and you want to continue to seem superior to them. But you feel like you're inferior to all of your colleagues, and you're afraid you're going to get thrust back in with the mob.
And it really depends on the field.
Yeah, that's true.
You see, in fields that are heavy on experiment, people will know if you're BSing at some point.
Generally, they'll figure it out. Yeah.
Yeah, because that's why you have materials and methods. Mm-hmm. And if they can replicate it, then they know you're telling the truth. And that's what this is where I laughed at. I was in disbelief when I asked Chris Thompson, someone I debated. There was a debate between he and I, R.N. Ron, Cindy Lincoln, October 5th, 2022. And he had me on his show. And the first two shows I complained. I said, dude, what's going on here? You're inviting me there to try to explain my theory, and you're not letting me explain it the best that I can. You're asking me all these irrelevant questions that are superseded by stuff. And I said, the next time I come on the show, I think it's only fair that I get to defend my viewpoint on my terms, because I have co-authors here, and this is not honoring their hard work. Mm-hmm. That you're querying me on just irrelevant stuff and trying to, it felt like he's trying to discredit our work. And I said, you let me argue this the way I think it should be. And I said, half and half. I said, you can disparage and criticize, but I don't think I should come on the show and just let you, run it the way you think and not let me bring the best evidence. That's not fair to me. And even worse, it's not fair to my co-authors. He said, okay. So I said, I'll present for 20 minutes, and then 20 minutes I'll grill you. You can do the same to me. We haven't had a show after that. I guess he, But I asked him, I said, do you think evolutionary biology should hold the same status as electromagnetic theory? And he said, absolutely. Look at all the departments they have. I'm just like, oh boy.
Did you realize that there were eugenics departments at universities at one point?
Hey, they're intersectional departments. They're all these other things.
Social justice departments and yeah.
Oh yeah. Oh my goodness. If I read some of this stuff that comes out of some of these postmodern these postmodernism inhabits. Like, do you really want to go that route? The size of the department legitimizes
Yeah.
The field. I said, electromagnetism, if it's wrong, there's no modern world.
Mm-hmm.
It has experiment. And that's the difference between some departments in academia that are experimentally focused.
Mm-hmm.
I have, as long as they're putting out good experiments and reporting the results honestly, they're going to be good. But anything that is driven by kind of like the herd mentality, that's going to, that's not a good environment. That's how you have these postmodern departments,
Yep.
Insular goodness. So anyway, I had a great time, Emory.
Yeah, that was a fun paper. It was a fun paper. I need to read the rest of it at some point. So that was a fun paper.
Yeah, that was. I hope Well, again, congratulations on your graduation from graduate school, and I know you're one to fill out your schedule, and but I hope we could do this again a little more frequently until you find something else to do.
Yeah, definitely. Just shoot me an email whenever you'd like to. I'm usually free in the evenings, later in the evenings, so at least currently. So let me know.
Look, Rebecca's here. Are you done with your stream, Rebecca? We were nerding out. Oh my goodness, it was so bad. If you were here, Emory, I'd be giving you high fives, man.
That was a fun stream. That was fun.
Oh, and Mark O'Connell asked, is Emory a doctor yet?
Not yet. I got to find myself a PhD program. I'm working on that.
Both of us, brother. So, hey, I asked Dr. Joel. I
suspect that Dr. Joel and I would probably end up killing one another.
Okay, but I was batting for you there, Emory. I asked him specifically.
I know. I know. So I suspect that we would probably not end up liking each other very much. So,
well, I got to admit, this was a fun stream.
Yeah, this was.
No, I think we could have even more fun if we just did on the route of discontinuity systematics.
If you want to just do a full stream on discontinuity systematics, I'd be down for that.
I'll be down for that too. But the thing is the way to make it fun is like the way we're doing it. We'll just say we could find evidence and say, take that, evolutionists. What do you think about that, Todd Wood? I'm sorry to pick on someone.
That's a Freudian slip right there. What we could do is Reed Remind's 1990 ICC paper.
Yeah. Well, I really feel I've stumbled on discontinuity systematics in the major protein families. You know why? I got it on good authority because R.N. Ra, I have him. He gave me permission to publish this in an email he sent me. He said, there aren't any flippin' common ancestors for all proteins. He used a different word. I just kind of, sanitized.
Yeah, I know what word he used because I know his profane tongue.
How's that for? Thank you, R.N. Ra. You've made the case for discontinuity systematics in major protein families.
Yep.
Oh, creation biology. Who? Do I know who you are?
Sal, look at the screen.
Oh, I saw okay. Oh, it's you. I was wondering who that brilliant guy was in the side chat. I said, am I being stalked by another creationist, by a rival creationist? Oh man, I got to do some house renovation tonight. So
I understand. Yeah.
Oh, that's your new.
Yeah,
Actually,
that's the that's the science channel. That's the new science channel.
Okay.
And there's an In His Image channel as well but that's just for social stuff. So that's
Okay. I gotta be honest here. For a fleeting moment, I thought when I used Todd Wood's name in vain that was him.
No, unfortunately, although that would be amusing, I don't think he stays up this late to watch our live stream.
Yeah.
Although I don't know if he has any kids. I've never heard him mention kids, so maybe he does stay up this late. I don't know if he has kids. I guarantee you he doesn't stay up this late.
Yeah. Well, brother, can we before we wander off backstage, I hope you don't mind if I have the closing combat music. Then we'll have.
That's fine. That's fine. Yep.
But this has been, Emory. This is really good actually, because I got to confess to you. As you know, I'm starting to now split time between physics and biology. I'll tell you one of the reasons why. There's a shortage of YEC physicists, especially with background in my the specialties of electromagnetism and relativity. I mean, I went to an ICC conference and and the more that I talked, I said, I think of all the creationists that I know, there are only 5 that have studied general relativity, and I was one of them. And that's oh my goodness, we're spread thin, because I didn't know it that well. And so I feel I have to, that's where God's calling me, because honestly, I think biology is in such good shape.
I would debate how good of shape biology is in, but we're in better shape than the physicists. We'll put it that way.
Yes, because it's plentiful. I'll tell you the main reason: the data. If we have some, just like what we did today, we could just read some of this and say, "Take that, evolutionists."
Mm-hmm. Yep.
And there's no shortage of that. It's only now, in the last couple of years with the James Webb Telescope, and now there's starting to be a climate critical of the Big Bang cosmology and just a lot of things. It's starting to break forward. So I've said it's not because of creationists that the creation science movement has moved forward. I had a conversation with John Sanford this morning, and he said, it seems like everything we need is just coming out this generation. I said, yeah, it's the glory of God to conceal a matter, glory of kings to search it out. But this generation, God has provided us stuff that is available to no other generation. We are in a privileged time. And greetings, Peter. God bless you. So we'll do discontinuity systematics. And I've said that's the way, that was the basis for the way Walter Riemann wanted to do baraminology. Mm-hmm. And I've said I'm not a baraminologist, I'm a discontinuity systematist, meaning I may not be able to identify the created kinds, but I think I can identify created systems and architectures. And that's very easy. I think the kind is, I believe they exist, but I want to be maybe a couple levels higher up, than an entire specific kind, maybe a general, architecture. So, I don't know how we can meld that with discontinuity systematics. With baraminology. But Walter was the one who pioneered baraminology based on discontinuity systematics. Mm-hmm. So we'll think it up, and I may get to see Randy. Guys, there's a 50% chance I'm going to Dallas, and I may get to go to the ICR. And hang out with the friends, and who knows if I get to have lunch with Randy Gugliuzza again, or I might talk to some of his associates that continuous environmental tracking. And I feel really bad. I'm overwhelmed with stuff, Yeah.
I think everybody in the field is, Sal. I've got more papers that I want to write than I could possibly shake a stick at.
Yeah. And well, brother, can we let me If you can, if you'd like to close us in prayer, let us give honor and glory to God and his great creation, like these telomeres and the Terra. Take it away, Emory.
Dear God, our Heavenly Father, I humbly come before you, praising and glorifying you for the day which you have given to us. Lord, I pray for, thank you for the time we've been able to share together tonight and the information that I have learned. This has been a learning experience for me. I've learned a lot about telomeres and subtelomeres and non-coding RNA and so on. More importantly, Lord, I get an appreciation for the way and the intricate design which you have used for your creation. And Lord, I thank you for the opportunity that you've given us to study your creation and to learn about it, so that we are able to understand how you made things and why you made them the way you did. And Lord, I pray that you will just bless the remainder of our nights tonight, help us to honor and glorify you as we go, and may everything that is said and done on this channel and on mine glorify and honor you. I ask this in the name of your Son, Jesus. Amen.
Amen. So, Emory, I'm going to meet you backstage after we close out with my favorite music. Actually, this is really for you because you liked it too. So, God bless everyone. Thank you for joining us.